This is a recording to the Introduction to Prion Disease talk, which was part of the Family Support Day on the 7th of September 2024. So we did things a bit differently this year in that we had 3 talks to start the day. So I started off by providing a very general background to prion diseases. So my name is Elizabeth or Lizzie Hill. I am a final year PhD student at the MRC Prion unit. You may also recognise me from previous events. I've been volunteering with the CJD Support Network for a few years now and have more recently become a committee member. My talk is going to be followed by a talk by Rachel Williams, who's a lead nurse at the National Prion Clinic in London, and she's going to be talking a bit more about the patient journey. And then to finish, Professor Simon Mead is going to provide an update to treatments. For those of you that don't know Simon, he's Deputy Director at the MRC Prion unit, also consultant neurologist and a committee member of the CJD Support Network. And just as a bit of context, we all work at the MRC Prion unit and National Prion Clinic, which is based at UCL in London. So before I talk about the biology of prion disease, I first just need to introduce a few general biological concepts. So I'm really going to use this as a basis to explain the biology of prion diseases. So we all have genes which you can think of sort of like an instruction manual that make you who you are. They govern everything from your eye colour to your height to how your body works. And you inherit genes from your parents. So half from your mother, half from your father. And genes are made up of a very special code, which we refer to as DNA. And this codes for RNA, which you can think of sort of like a messenger that makes the final product, which is the protein. And you can think of proteins like the workhorses of the body. They do everything from fighting off infections to running your metabolism to make sure that your body is working. We have many different proteins in our bodies. We actually have more than 100,000. And these proteins make up cells, which are the building blocks of the human body. And proteins can be broadly classified into 2 main subtypes. Some of them have more structural roles and others have more functional roles. And one important concept to note about proteins is that the correct folding and structure of a protein is absolutely essential for it to be able to perform its job. So when a protein is initially produced, it's in this unstructured form. And then as it starts to fold, it takes on these basic repetitive structures. So this beta sheet here at the top is one example. And this coil-like structure at the bottom, which we refer to as an alpha helix, is another. And this is all along the pathway of the protein forming its final 3D structure. And it's this final 3D structure which is essential to allow the protein to be able to perform its job. So now I'm going to move on to talk a bit more about the biology of prion diseases. So as I said, we have many genes, and one of these genes encodes the prion protein. And we all have this gene. It's completely normal and it's passed on from our parents. This gene codes for RNA, which makes the final protein, which in this case is the cellular prion protein, otherwise known as PRPC. So what does PRPC do? So in short, we don't know. PRPC has been suggested to have an array of important functions, especially in the brain. However, none of these have been definitively proven. A couple of examples of this is it's been proposed to protect brain cells from damage. It's also been proposed to allow brain cells to communicate with each other, so we believe its function is important. However, in prion disease, this normal form of the prion protein, PRPC, is corrupted to form a misfolded disease-related form. So in this diagram, you can see normal PRPC on the left here in its properly folded state. You can see that it's rich in these coil-like structures, which I described before as being alpha helices. But you can see on the right here that in the disease, it has a completely different fold, and it subsequently stacks up, and it's this form which causes the disease. And how it's able to cause the disease is that this is a self-amplifying process. So here I've simplified things. So PRPC is represented by a grey circle, and the misfolded form of the prion protein is represented by an orange hexagon. And what happens is that when this misfolded form comes into contact with the normal form, it causes the normal form to misfold. You then get these fibrils, which then fragment, and it creates this self-amplifying process. And what this essentially means is that you can start off with just having one or a few misfolded forms of the prion protein in the brain, but because of this self-amplifying process, it's able to completely overtake the brain. So now I'm going to just go through the different types of human prion diseases in a bit more detail. So there's three main types. By far the most common is sporadic CJD. This accounts for around 85% of cases. Then there's the inherited forms of the disease. These account for around 10 to 15% of cases. Then finally, there are the acquired forms of the disease, which account for less than 1% of cases. So I'm going to go through each of these different forms in more detail in a couple of slides. But first, I just wanted to talk you through a few defining features which unite all three of them. So they're all neurodegenerative diseases, which means that brain cells die and underlie them. They're all progressive in nature, meaning they get worse over time. And they're all fatal, with there being no established disease-modifying treatments, although Simon will go on at the end to talk about some up-and-coming therapeutic options. All of them have the misfolded form of the prion protein at their core, and they all have this underlying disease process whereby the misfolded form is able to cause the normal form to misfold, creating this self-amplifying process. Broadly, they all have the similar pathological hallmarks. So if you look at brain tissue under the microscope, there's a couple of stains you can do. So at the top, this is spongiform change. So here the brain tissue is stained in pink, but what you can appreciate is that there's these holes in the brain tissue. This makes it almost appear like a sponge, given the name spongiform change. Then at the bottom, this is a stain that's stained specifically for the misfolded form of the prion protein. And what you can appreciate here is that there's these brown deposits, and these are accumulations of misfolded PRP, and this is seen in most forms of prion disease. So now going on to the different types in a bit more detail. Well, sporadic CJD is, as I said, by far the most common. It accounts for around 85% of cases. It generally affects people between the ages of 45 to 75, with an average age of onset of 65 years. The disease duration varies, but it's generally rapid of onset, and the disease is usually measured in months. So what causes sporadic CJD? Well, we think that it's caused by PRPC spontaneously misfolding to a misfolded form through mechanisms that we still don't quite understand. But as I said, we have many, many different proteins in our bodies. We're constantly synthesizing them. So there's always potential for an error in this production process. But what we do know is that there's certain risk factors which can increase the risk. So one of these is age. We know as people get older, there's an increased likelihood of sporadic CJD occurring. And there's also certain genes which can increase the likelihood of the disease occurring. And that's actually the area of my research. Although I would like to stress here that these genes don't cause the disease, they just very subtly increase the risk of disease occurring. And then because of this self-amplifying process, this is able to result in the disease. Then there's the inherited forms of the disease. So here there's actually an error in the DNA code of the gene which encodes the prion protein. And this abnormal gene therefore encodes a abnormal protein. And in this case, we think that PRPC is more likely to misfold. So the inherited forms of prion disease are passed down from parent to child. So as I said, we have 2 copies of each gene. So if one parent has an error in one copy of the gene, there's a 50% chance of this error being passed down. So essentially what this means is that on average in any generation, around half of the children will be affected. There's many different types of inherited forms of prion disease. Three of the most common is GSS, FFI, and familial CJD. It's estimated that there's around 1,000 pathogenic mutation carriers in the UK. And really, the age of onset, time course, and symptoms depend on the nature of the specific DNA error. Then the final form of human prion disease are the acquired form of the disease. These are caused when an external source of preformed prions is introduced into the body in a very specific set of circumstances, and this can then cause a normal form to misfold. So for the purposes of today, I'm just going to focus on two main types which are most relevant to this audience. The first is variant CJD. This was caused by exposure of individuals to infected food products. So this was heavily centered around the BSE epidemic in the 1980s and 1990s, where cattle feed became contaminated with a source of prions. It typically affected younger people with a relatively longer time course. However, since then, feed regulations have been put in place and also public health measures, and these seem to have been largely effective. Then the other form I wanted to talk about was iatrogenic CJD. This is caused unintentionally by a medical procedure. So some examples of this. So historically, children with growth defects were administered human growth hormone, which was derived from human donors. This became contaminated with a prion source and resulted in the development of iatrogenic CJD in some individuals. However, now growth hormone is produced synthetically in a lab with no risk of transmission. There's also been instances where iatrogenic CJD was caused by implants of dura, which is the membrane which lines the brain. However, again, now this is produced synthetically in a lab with no risk of transmission. There's also been rare instances in the past where iatrogenic CJD has been caused by blood transfusions. However, this is very specific to blood donors that had variant CJD. And also there's been measures that have been put in place that make this not very likely. And then finally, there has been rare instances in the past where iatrogenic CJD has been caused through surgical instruments in very specific circumstances. However, again, there's measures that are in place that make this very, very unlikely, and it hasn't been reported for a long time now.